The chapter Molecular Bases of Inheritance is one of the most concept-heavy, yet high-scoring chapters in Class 12 biology. NEET consistently asks multiple questions from it. In this video, let's go beyond quick revision. We'll see how to study, what to revise, and which areas to focus on to maximize your marks. Start with the experiments that proved DNA is the genetic material. Griffith's experiment and transformation principle. Avery, McCarty, where DNA is the transforming principle. Hershey-Chase experiment. A classic blender experiment with bacteriophages. NEET often asks direct fact-based questions here, like which isotope labeled protein, S35, and which labeled DNA, P32. Here is a quick comparison table: DNA versus protein labels, isotope used, and where detected in Hershey-Chase, for quick revision. Next, revise the Watson and Crick model of DNA. Focus on double helix and antiparallel strands. Base pairing rules like A equals T and G is identical to C. Then, hydrogen bonds, 2 between AT and 3 between GC. 10 base pairs per turn, 3.4 nanometers per turn. NEET questions often ask about base pair length, number of nucleotides, or comparing GC versus AT bonds. Also revise types of RNA, including mRNA, rRNA, tRNA, along with their functions. You will find all these questions to practice on extra classes. There are 6 tests, with 400 plus questions, including those from the previous year exams. Don't skip DNA packaging. In prokaryotes, DNA is organized with polyamines in proteins, while in eukaryotes, DNA wraps around histones forming nucleosomes. Questions may ask about nucleosome structure, histone octamer with H2A, H2B, H3, and H4, with H1 as linker histone. DNA replication is a high-yield area. Understand the process and enzymes. Helicase unwinds, primase lays down RNA primer, DNA polymerase synthesizes, and ligase seals Okazaki fragments. Some of the key points are: replication is semi-conservative, proved by Meselson and Stahl's experiment using N15 and N14 isotopes. It occurs during the S phase of cell cycle. Also revise leading versus lagging strand differences. NEET often asks enzyme functions or identifies semi-conservative replication evidence. Transcription is the synthesis of RNA from DNA. In prokaryotes, a single RNA polymerase transcribes all types of RNA. In eukaryotes, RNA polymerase 1 makes rRNA, 2 makes mRNA, and 3 makes tRNA. The steps are initiation, elongation, and termination. Know the difference between coding strand, that is, sense, and template strand, which is antisense. NEET loves asking, which RNA polymerase synthesizes mRNA in eukaryotes? Another scoring area is the genetic code. Key properties are triplet, universal, non-overlapping, and non-ambiguous. Out of 64 codons, 61 code for amino acids, 3 are stop codons, which are UAA, UAG, and UGA. Start codon is equal to AUG, which codes for methionine. NEET often asks codon-based MCQs, like which codon codes for stop or start. Translation is the process of protein synthesis. Ribosomes are the site. tRNA is the adapter molecule, carrying amino acids. Translation steps include initiation, elongation, and termination. Elongation requires GTP for energy. Pay attention to P site and A site of ribosome, and the role of peptide L transferase. NEET may ask questions like, what is the role of tRNA? Or which energy currency drives elongation? Focus on lac operon in prokaryotes. Lac operon is an inducible operon. Structural genes are Z, Y, and A. A regulator gene produces repressor protein. In the presence of lactose, the repressor is inactivated to genes transcribed. NEET often asks, what is the inducer in lac operon? The answer is allolactose. Two small but frequently asked topics are Human Genome Project, completed in 2003, identified 3 × 10⁻⁹ base pairs and approximately 20,000 to 25,000 genes. DNA fingerprinting, based on polymorphism in variable number tandem repeats. Lastly, expect simple factual questions here. From this chapter, NEET usually asks, match the following, or match enzymes with functions. Fact-based questions including codons, base pairs, and RNA polymerases. Concept-based questions on lac operon regulation and semi-conservative replication. Calculation-based question like, how many base pairs in a given length of DNA? So, while revising, ensure you practice NCERT examples and past 10 years NEET questions on extra classes. There are more than 400 questions altogether in 6 tests. The link is in the description. Here's a quick revision strategy. Memorize experiments and scientists with outcomes. Prepare a table of enzymes with their roles. Revise codon chart daily, stop codons, start codon, and degeneracy. Practice PYQs and NCERT-based questions, many are repeats. Don't ignore small sections like HGP and DNA fingerprinting, they give easy marks. Students often confuse template strand with coding strand. Forget that AUG codes methionine. Mix up RNA polymerases in eukaryotes. And ignore lac operon details. Avoid these errors. NEET thrives on these confusions. So, to master molecular bases of inheritance for NEET, revise NCERT line by line. Focus on diagrams, especially Watson-Crick model, transcription unit, and lac operon. Memorize codons, enzymes, and scientists. Solve NCERT back exercises and previous year's questions on extra classes. With this approach, this chapter becomes straightforward and a guaranteed scoring area in NEET. All the best for your NEET preparation. Join extra classes for free today. Use the promo code in the description.